Today I am going to give a brief presentation about my work during the last 5 years. As you see here, Human-Computer Interaction Principles for Developing Web-Based Genomic Resources is the title of my PhD. As the title suggested, the main goal of my PhD is to improve the bioinformatics tools using human-computer interaction design and visualization techniques. To apply the design principles, we need to have a database and the initial infrastructure. Therefore, we developed a database system by integrating biological data. Then develop tools using different design principles and finally evaluate them with various usability methods. These are the content of this presentation. Why we need bioinformatics tool. Development of specialized database. Then I will discuss briefly about integrating external web resource or database into Plangini. A case study to show how to use our tools. Finally, let's look at how to apply human-computer interaction design principles to improve the usability of our tools. Let's begin with why we need usable bioinformatics tools. This is quite interesting because the scientific method we use in science is quite similar to the process that we use in the design. In both processes, we do not know the final end product, but we have an idea how to get there. If you are a scientist and if you create a scientific figure with a lot of data, it might be confusing to the audience. On the other hand, if you are a designer and if you create a tool, it might be pleasing to the audience with a lack of information. However, if we could combine these aspects together, we can deliver a clear message to the audience. This is what I mean by the sweet spot of visualization. Scientists have predicted a continuous increase of data growth for next 10 years. This figure shows the future data increase. The future data increases using three prediction methods. The red line is based on the historical data, while the blue line is based on the Moore's law or Moore's principle. The yellow line represents the Illumina prediction. This emphasizes that we need more resources to store, filter, manage, analyze those complex data. As volume and complexity go, the current databases and the database resources are not sufficient to cater the needs of the research community. The first nucleotide database started in 1995. Then different databases known as database archives and specialized databases have been established during the last 2 decades. From 2010, more focus on expression atlas genome variants, it's keep growing. If we look at the plant-oriented databases, the first forestry species, Populus trichocarpa, was sequenced in 2006. By 2018, there were 148 plant reference genomes and only 52 tree species shown in this diagram. Simultaneously, there are corresponding databases or specialized databases were established during the last 2 decades. For example, Plaza tree gene Arabidopsis. So having or developing a specialized database system is important. Now take a look at how can we develop those specialized database systems. In Plangini, we establish a specialized web resource called Plangini, which is Plant Genome Integrative Explorer, to search and explore genomics and transcriptomic data for different plant species. If you think Plangini as an automobile, we would need well-structured and scalable core system to run Plangini. Earlier version of Plangini was based on Drupal content management system. Drupal is a content management system for making professional websites. However, we completely rebuilt a new Plangini with a dedicated system called Genome Integrative Explorer System. Genome Integrative System facilitate Plangini as well as external group in setting up their own web resources for transcriptomic and genomic data used in Project Raw data like GFF3 FASTA. The system contains four basic components. The main Genie system, core plugins. User can easily convert their tools into the plugins. It also possible to change the entire layout using different themes. The entire system is free and open source, can be downloadable. When we think about the database, ideally we think the database is just simple storage of data with a single user interface. But the reality is far more complex than we think, as shown in this figure. Here I will briefly explain the basic architecture diagram in Plangini based on the Genie system. We are getting data from different data sources like SRA, ENA. By using different scripts, we convert those data into suitable formats and stored in Plangini databases. Then we have different services to interact with our tools. Gene Search tool is the starting point or heart of the Plangini system. User can easily create new gene lists by typing gene list IDs, synonyms or description. It allows us to search results into a gene list. User can use the analysis tools such as genome, expression or comparison tool to do further downstream analysis. The tool results can be visualized in our Plangini websites. It also has external links to other resources like Kegger, Thea, Python. And we recently integrated parser pipeline with Plangini. So all the green line in this diagram shows the external components and the blue line represents the components or the items from the Genie system. As you see here, we can easily separate the entire system into five main components: data sources, internal storage, web sources, centralized tool, and web resources. Because of the Genie system and the recent modification to the entire architecture, we added few more Genie sites into Plangini platform. We hope the number of species will grow in the future. Here are some of the users in Sweden, South Africa, Belgium, China, and France who are using our Genie system to make their own web resource. Now, as I mentioned before, we use Plangini and Plaza with Plangini database. Plaza is a plant-oriented online resource for comparative evolution and functional genomics. The current Plaza includes more than 70 species. The Plaza platform consists of multiple independent clades focusing on different plant clades. We consider this as a good opportunity since we already established one of Plaza instance to one of our collaborative projects. We consider this as a good opportunity since the Plangini we have expression and annotation data, and in Plaza we have a lot of ortholog and gene family information. When we combine these two datasets and tool together, we can create nice tools for the new Plangini. In Plangini, we have our own database structure, and Plaza there is quite different database structure. As our plan proceeds, we need to combine both Plangini and Plaza databases, which is similar to the translation translating of Swedish word to Dutch or Flemish words, or in our case, it is similar to moving between Aspen to Arabidopsis. Plaza uses mainly four different methods for identifying orthologs. As this figure shows, selected popular genes has three homolog genes in grape. All four methods are agree with first grape gene, and only one method or only one method are in agreement with other two genes. Here is an example how can we use this information with plant gene to integrate cross-species movement between different sites. We have different gene sites for different species, but we do not know how to move from one species to another. We can connect different gene lists using cross-species ortholog genes. So we can use Plaza ortholog information. As I mentioned earlier, Plaza has four basic methods to calculate ortholog. So we can use this information to move between sites. I will here show you how can you use in real world website. Now we are going to create the cross species gene list using best diamond hits. All I need to create a gene list. Here I type lactase to find lactase related genes and save it as a gene list. Then I will go to the cross species button and click best diamond hit to create best diamond cross species gene list in popular fruits and Arabidopsis websites. When we go to the corresponding website, we can see the best diamond gene list. Here is the current version of planting tool. We integrated so many species. We have a tool called Explorer. Explorer is a tool where we can see most of our pagination tools in one interface. The user can easily add or remove tools to the Explorer and also create cross-species gene list or move between different datasets. Currently, Explorer is working with ASPLIB, but we are planning to integrate with ASPUD and node datasets. Here I will show you a quick demonstration how this tool works exactly. Here I type CESA to find cellulose synthesis genes. Then I click OK. Then it will save as a separate gene list. Then we can add different tools to the Explorer by clicking different tool icons. Here I add Ximage, Exploit and XNet tools. When we click the line in the Explorer, we can see the simultaneous changes in other tools. It is also possible to rearrange and move or play around with tools, or remove tools and add new tools. There is also possibility to move between different datasets. We can also change the gene list. Now I am going to show you the recent case study based on our tools. Lactase and peroxidase are involved in lignification. It is hard to find specific genes because of the size of the gene families. They are big. But we need to find specific eucalyptus genes that are involved in secondary cellular formation using plant gene tools. Maybe you have an idea. I will show you what we did in our paper. We used this publication and found Arabidopsis lactase genes. Then we get homologous genes in eucalyptus using our BLAST tool. We found 44 lactase homologous genes in Eucalyptus grandis. We also found 93 class 3 peroxidase genes in Eucalyptus as stated in this publication. We combined both together. All we did was open the gene list and create a new gene list using these 137 genes. Then we go to the XNet tool. XNet is a tool where we can see the expression network in given set of genes. Circles or nodes represents the genes and the edges represent the co-expression between two genes. Color of the node represents whether they are highly expressed in a specific tissue. There are options to expand the network to find neighboring co-expressed genes. Like this. Here we have 137 genes, but we cannot see significant correlation between each other, or we can't say clear clusters. Therefore, we are going to expand the network. Here I will select all the genes in this network and click the expand button in threshold 6. Then I can clearly see two subnetworks. In one subnetwork, we can see 51 genes from 137 genes and the 8 genes from the 137 initial genes. Then we will further investigate this subnetwork 1 and subnetwork 2 with other tools. We move to X heat map, where we can see the heat map for selected genes and selected samples using hierarchical clustering. According to the X heat map results, we can see most of the genes in subnetwork 1 are expressed in roots and subnetwork 2 are expressed in immature xylem. Now we move to the enrichment tool, where we can see the enrichment results based on gene ontology. According to the enrichment results, subnetwork 1 are enriched in stress-related genes, where the subnetwork 2 are enriched in cell wall formation. Now we have an idea which genes, in this case subnetwork 2, are involved in cell wall formation. Therefore, we selected the genes in subnetwork 2 and continue our investigation. Now we look at our candidates gene in X-image, where we can see the expression variation among different tissues. As you see here, most of the genes are highly expressed in xylem tissue. Also, the co-expressed genes are involved in secondary cell wall biosynthesis like cellulose, hemicellulose, or lignin. The advantage of having plant gene sites is we can do cross-species comparison. Here we investigate the starting candidate gene in subnetwork 2 with Arabidopsis and Hoplodopsis. This further confirms that our hypothesis, because of the orthologous network of other species, are also involved in secondary cell wall. And this making this database and integrating with external resources help and contribute to the FAIR guideline. FAIR guideline is a guidelines that scientists can follow to make it findable, accessible, interoperable, and reusable. In this, we come to the last part of our presentation, applying design principle to improve the usability. I use some design principles to improve the usability of our tools and validate if these design principles are worked well using different usability methods. These are the design principles that I used during my study. Affordances. Affordances are the relationship between the object and the entity. In this case, and the tool and the user. Signifiers. Signifiers are the signal communication devices. These signals are tell you all possible action in that function, or in this case button, or it can be the error message. Feedback, sending back to the user information about what action has actually happened in the backend is important. Then user can decide whether they need to stay in the tool or move to another step or do something else. Mapping, we try to use real world metaphor for like bugs or leaves in our tools. So we can easily relate the real world metaphor with the virtual world metaphor or the functions. So user can easily identify these functions. And we, as you may know, 8% of the men in the Europe are colorblind. So we try to use colorblind color palette when we develop our tools. This is important because then user can easily see the color distinguish between, for example, in expression variation. So there are different constraints like physical, semantic, cultural, and logical constraints. As I said, the affordances, if you look at this figure, the strong to weak affordances shown in this figure. As you see here, the first button, it gives us strong affordances where user feel to click that button. When it comes to the weak affordances, user really don't feel anything. Clear, meaningful buttons, labels, notifications, and icons are helpful to understand the possible actions. These signals are called signifiers. Affordances give a clue about the possible actions, while the signifiers help to communicate where the possible action should take place or help to discover other possibilities. When we want to show an expression variation among different issues for a given gene, of course, we can use a table like this. But it is quite hard to identify which tissue is highly expressed or which tissue is lowly expressed. However, if we show these expression values in a figure like this, this may give a better understanding about the expression variation, but it is hard to distinguish colors. However, if we show the expression values by sending the colors in different samples, we can easily see the variation. This is because the patterns of colors are more easily understood by the human eye as a means of interpreting data than the corresponding numerical value. So we use the same principle in our tools. We try to use the patterns, colors as much as possible. Flexibility trade-off. Let's look at iPhone and the old phone. The new smartphone facilitates us to do like taking pictures or sending emails, sending text messages, or of course making a call, and so many other features. When you look at the old mobile phone, we probably can be able to make a call or perhaps can be able to send a text message. So when we add more features to the device, the flexibility increases, but the usability reduces because user can't figure out what should I do with this much of functions. For example, in this tool, you are familiar with the word processor with a lot of functions. Most of the users are confused or pretty useless to having this much of functions. The same principles apply to our tools. When we add more functions to the tool, the flexibility of the tool increases, but the usability, the how can user use these functions, is decreases. So we wanted to find the balance. Therefore, we try to use the layering principles. For example, we group the similar categories into the one layer or one group. So user can easily identify these components or the similar functions. And also we try to reduce the cognitive overhead of the user by using single window, where user can find most of our tools in one single window. And also we try to use iconic representation, where user can see the respective icons and interact with them without giving so much information, which perhaps can call the information hiding. Human search for information is similar to the way that wild animals hunt for food in nature. Let's take an example. We have a forest with a lot of rabbits in it. There are two areas called patch A and patch B. Let's say there are 4 rabbits in patch A and 2 rabbits in patch B. Where should the fox go and hunt? Clearly, the fox go and hunt in patch A because there is more game. Now after a while, let's say fox has caught and eaten 3 unfortunate bunnies and there is 1 lucky one left. There is 2 left in patch B. What will the fox do? Keep hunting that last rabbit in patch A or go to the richer hunting grounds. The fox should go to the richer hunting grounds. That is the fact, what biologists find when they study these things. Similarly, we can do our usability studies. We should not hunt until every last usability problem in version 1 of our design. Rather, we should stop after a while, and now it's time to move to a richer hunting grounds. In this case, version 2, to discover more rabbits, because hopefully we have successfully identified removed some of the key issues in version 1, meaning that users now can deploy and engage with the user interface and do more interesting user behavior. That leads us to discover new interesting usability problem or new rabbits. Any usability problems that was present in version 1 of the design was not found and therefore not fixed will still be in the version 2 of the design to us to discover. That's one case we are better off than the fox, because any rabbit that was surviving in patch A is not going to follow the fox or to the patch B to be hunted there, because only a suicidal rabbit can do that. So we are better off than a fox. We should follow this principle. So not hunting to extent in one area when there are other things we can do and spend our resources that will lead us to better results and better freight. So we use the version 2 of our design. This is quite interesting. You have seen this many times in your life. Even though there is a clear road exists, people use their social innovating abilities, skills, to create their own shortcuts. We use the same metaphor in our tools. Examples of we investigate the user behavior using different analytics tools. Based on the user behavior, we place the essential components of our tools, which is similar to the desired line of following the user behavior. As a summary, first start with the prototype. Our goal is to make the production version of the tool. Along the way, we follow these steps. First, apply the design principles and visualization techniques and make some changes. Then, analyze our tools or prototype with different analytics tools and make some changes, and then get feedback from design experts and again do some changes. If the real world users are happy, we make it as a production version, otherwise we do this iteration one more time. With this, I cover all the topics in my presentation. These topics are actually related to the publications that I have mentioned in my thesis. With this, I hope I gave a brief overview of everything, and thanks to my supervisors and my team for the support and motivation, and all the amazing people at UPSC and the Institution of Informatics, and thanks for listening.